Welcome back to the Old Turtles podcast, the show about the future of work, the future of health, and the entrepreneurs who are building those futures with new tech. I'm Phil Libben, the CEO of Old Turtles. Today I'm interviewing Arun Wiita, who runs a lab at UCSF that's discovering new treatments for blood cancers using emerging technology. I'm here with Arun Wiita. Arun is an associate professor at UCSF, and he heads up a lab that focuses on discovering new treatments for blood cancers and genetic diseases using pretty cutting-edge technology, CRISPR, machine learning, next-gen sequencing, stuff like that. Thanks for being here, Arun.
Yeah, thanks for having me.
So that's a lot of exciting-sounding words. Maybe we start with just unpacking what some of those words mean. Let's start with blood cancers.
For sure. So essentially, when it comes to cancers, we think about two main categories. There's both solid tumors. That's what you may think about: breast cancer, prostate cancer, lung cancer, colorectal cancer, some of the most common ones in the U.S. Blood cancers, we think about two major types: leukemias and lymphomas. Essentially, these are when the blood cells in your body that are normally produced to help fight off infections, for the most part, really run your immune system, they can go awry. So they start growing out of control. There are many different ways that can happen. And essentially, some of them can circulate in your bloodstream. Those we call leukemias. Oftentimes, those are more aggressive, more deadly. Some of them can grow within your lymph nodes, which are parts of your body that essentially process those foreign antigens, things coming from bacteria, things like that. Some of those cancers can grow within the lymph nodes. Those we call lymphomas. But essentially, there's a broad range, and there's a few that we specifically focus on in my lab. We like to look at ones that are derived from so-called B cells. These are important cells, again, for handling foreign invaders, essentially, in your body. And again, these cells can go awry. One we focus on specifically is called multiple myeloma. It's diagnosed in about 30,000 patients or individuals a year in the U.S., and right now it still doesn't have a cure. So that's a big part of what we do in my lab.
Let's kind of take a holistic view about this. There's a few different areas that are probably worth talking about. There's, I guess, prevention. How can you make it less likely to develop a serious cancer? There's diagnosis, and there's treatment. How have changes in technology played into each of those three?
Yeah, so I'd say the first one in terms of prevention, I think really has gone hand in hand with diagnosis in a lot of ways.
You want to get things diagnosed as early as possible, I guess.
Exactly. So that's really where a lot of emphasis has been put in the past few years. Because when it comes to prevention, honestly, I think you see this week in the news in terms of now we don't know if red meat is necessarily that bad to eat anymore, right? Which is like a study that just came out yesterday, and it was in Internal Medicine and caused a big sensation. I mean, I think when it comes to prevention, the one thing we've been really good at is telling people to stop smoking.
Right.
Like that has been, because smoking influences development, not of every cancer, but many, many types of cancer. So that's the one thing we've been really successful at.
Right. And stop licking like the radium watches or something.
Exactly.
We kind of got that done out of the way in the 50s. And then,
Exactly. So avoid the toxins, avoid things that are clearly toxic and carcinogenic, avoid smoking. But, besides that, we haven't, I would say, from a public health perspective, made a ton of progress on prevention. So right now, I think a big focus and where a lot of investment's going and a lot of academic and commercial research is in early diagnosis as a means to prevention.
Right.
And so
because the treatments are much more effective or cheaper earlier on.
Exactly. That's the hypothesis. I think the big question when you start doing really early screening, early diagnosis, we also know that many individuals are going to have something that looks like a cancer that never actually develops into something deadly. And these are things actually primarily our immune system is able to clear.
Right.
And so that's one of the big questions when we start getting better and better at detecting very early stage cancers is, are we going to be treating patients, doing interventions that they never needed in the first place? So I think that's going to be the next really big question we have to answer once we get these technologies robust enough to detect them very early.
Right. So then let's talk about detection, about diagnosis. You just hear in the, I think everyone has a family story or hears from friends about someone who was diagnosed with a cancer and is kind of amazed how quickly it moves. You have a friend who was diagnosed relatively recently, and then within a matter of months, it's a very serious condition. It always feels like a lot of these diagnoses they're just happening really, really late. And a lot of the diagnoses that you hear about are kind of accidental. Like, I went in for some pain and something, and all of a sudden it turns out you have late stage cancer. Why is that Like, why is that what seems to be the norm in the industry? Why are we kind of waiting for a while, and why are we sort of guessing at what things are?
Yeah, I think the main issue right now is that, we've been working for a very long time to try and come up with good screening tests for cancer. But the issue is that
before there's like symptoms, right?
Exactly. So the optimal situation, what everybody wants is you go to the doctor once a year, get a blood draw. Or maybe you don't even go to the doctor, you go to the Walgreens and get a blood draw.
Or you wear a wearable of some kind.
Exactly. And even now, moving toward wearable. Exactly. So you have your, my disclosure, my wife wears Apple. So I got to say, you have your Apple Watch. It would tell you if you have cancer, right? Yeah. So that would be the optimal situation. So I think that's where everyone would like to go. The main issue we found, at least historically, though, is the few markers we've had that we can detect in the blood that could be indicative of potentially having cancer. They run a lot of these risks of overdiagnosis. And that's actually what we've seen for mammography too. So we've seen that that's another non-blood related biomarker, but radiographic related. But we've seen the same controversy about, do we have too much overdiagnosis or too many women having mastectomies or other treatments they don't need? We see the same thing with prostate cancer exams. So I think that's that's going to be the critical question is, can we have something? So my clinical training, I'm a molecular pathologist. And so what my training was all about is all laboratory-based testing. And the main thing we think about is both sensitivity of a test and specificity of the test.
Right.
And so if it's 100% sensitive, that means you're going to detect everybody who has the condition. You're not going to miss anybody.
Yep.
If something's 100% specific, that means that everyone you say has that disease based on a test actually has it.
Right.
But the problem is that it's very difficult to get a test that's 100% sensitive and specific, if not impossible. But that's really what you want in one of these screening tests.
Right.
And so historically, that's been the major challenge of going on a single protein biomarker in the blood. The big question now and the big emphasis, what we've seen in early cancer detection, at least, there have been a lot of companies, say like Grail is probably the most famous in the space, raised a lot of money, spun out of Illumina, who's the giant in next generation sequencing. They were really focusing on looking for mutations in DNA that are indicative of cancer.
How much of this is improved by going from kind of a one point in time test to looking at changes and trends over time?
That's a really good question, actually. There's a really cool study that recently came out of Stanford, actually, where they kind of had a dynamic risk assessment, and not necessarily for detection, but I think you could apply the same model, where their hypothesis, they actually used the analogy they used was if you're, say, watching the Super Bowl, and you see at the beginning of the game, each team has a 50% chance winning, it's 54-46 or something. But then as the game goes on, someone scores a touchdown. Now the percentages have shifted quite a bit. Team scores another touchdown. Now it's one team's 90% versus 10%. Can you have something similar for someone who's treated with cancer where after the first round of chemotherapy, you see these certain biomarkers in their blood go down? Well, now they have a much better chance of being cured. Then after the next round of chemotherapy, you see these different markers change. Now you're 90% certain they're going to be cured. And some patients will be the opposite. I think we could see the same thing for early detection if we have these longitudinal measurements. How things change over time. We could definitely feed those into similar models. We just don't have the data yet for the most part.
And are blood cancers any harder to diagnose? Because, and I'm just completely guessing, but there's like less physical stuff to see? Like there may not be a visible tumor as much?
That's a great question. I would say that
it's probably not a great question.
No. It's interesting because it's because it's so overall, blood cancers are less likely to cause like physical pain, I would say, in one location, just from the mass of the tumor. Oftentimes, that's a big problem. Some tumors that tend to get diagnosed very late, at least for solid tumors, are ones where it doesn't bump up against other nerves or other areas that cause a lot of pain. Blood cancers. Oftentimes, they can be in the similar situation where they could be growing what we call indolently, very slowly for a long time. You wouldn't notice anything. And then suddenly they just kind of explode. And then you're very, very sick all at once. But sometimes they can lead earlier on to fevers, things like that, like kind of unexplained, maybe non-specific symptoms might be enough to get you to the doctor and get you checked out. So we kind of see it both ways. Like even the cancer that we study the most, multiple myeloma, that often gets found kind of incidentally, or at least the precursor stage of it, where it hasn't developed in the full-blown cancer yet. But we can see that you're kind of on the way, but not everyone's going to develop it. So we have this early detection, but right now we don't have enough evidence to say we should treat those people found very early.
Right. So what's some of the work that you're doing to get detection to be as early as possible and as I guess specific as possible for blood cancers?
So in our lab, we're not doing so much early diagnostic testing, but there are a lot of groups who are working on that. And I would say that one of the biggest, as I mentioned, kind of Grail's initial approach was to look at just DNA-based signatures. And this is something that people have been trying to look at, basically look at circulating DNA in the blood. We know that all cells in the body, we like to think about ourselves as just being alive, but it turns out that we have millions of cells every day that are dying, but they're being regenerated also. And so tumors also have this cycle. Even if they're growing really rapidly, a lot of them are dying at the same time. And so they actually release DNA. And so that goes around, circulates in your bloodstream. And so the initial thought was, if we can find that DNA with these mutations specific to tumors, that's going to give us these really powerful early diagnostics. What ended up happening is that it turns out that some of those mutations are found in normal tissues too. And it's really hard to distinguish, is this from the cancer or is this from normal tissue? So now the cool advance that's being made is looking at modifications to DNA and specifically something called methylation. So you basically put on like one little carbon with 3 little hydrogens on this one base in DNA. That can actually have thousands of different modifications across DNA, and they can actually be very specific to tissues and to cancers. And so most of the companies doing this early detection are moving in that direction of, instead of just having maybe like 10 or 15 or 20 mutations you can look at thousands of methylation sites. And so I think that this and these patterns can be much more specific. So I think that's where the kind of next big advance, also combining with other modifications, either of proteins, modifications of RNA. I think that's kind of where the next steps are really going.
When you say you can look at thousands of these sites, by you, do you mean a doctor or do you mean an AI?
So that's a great question. I mean, I would say first is going to come down to the initial technology. Most of these are going to be next generation sequencing based when you're talking about DNA or RNA. And so just to relate what that is, essentially the idea behind next generation sequencing, we call it next generation because historically, if we wanted to take the sequence of DNA, again, this is the genomic code, we have it in all of our cells. Typically in the lab, we might want to look at a short stretch of that DNA and say, we have 4 bases, A, C, T, and G. But the order of those bases is incredibly important for determining what that DNA is doing.
Yeah, kind of matters. Yeah.
Exactly. And so historically, we would take this very kind of laborious biochemical process called Sanger sequencing to tell what one sequence of DNA did. Now we have this technique called next generation sequencing, been widely used for the past 15, 20 years, where essentially we can sequence millions and millions of DNA molecules at the same time. You generate a lot of data. Historically, it hasn't necessarily been analyzed via AI type approaches, but that's become much more common in the past few years. And so when we look at DNA and RNA, we're going to generate these large data sets. We can either put them through kind of standard informatic pipelines where essentially we try and say, where in the genome did these sequences come from and do they have alterations versus a reference genome? That's kind of the standard way to do it. But the other way that is becoming more common now is to use kind of either machine learning or other type of classifiers to say have this big cohort of patients, a big cohort of normal people. Here are these altered signatures I find in the patients, I don't find in the normal individuals, and I can use some type of AI framework to build a classifier to say, here are the signatures. I might not understand the biology behind all of it, but here are the signatures that are indicative of that cancer patient or whichever disease.
You know, we had another guest on a few months ago. It was Dr. Katerina Sofia Volz. She's the CEO of a startup called Occam's Razor that's doing AI for Parkinson's disease discovery. And her kind of main hypothesis, which I really think is fascinating, is she's kind of saying we're sort of at the tail end of this like heroic age of medicine where breakthroughs are made by, great men through some insight or some accident. Like you have a Eureka moment. You wake up and you're like, aha, it's a double helix. Or, there's an accident and you leave some bacteria out in a petri dish and you get penicillin or something. You're just kind of saying like the things that were simple enough that you could have had a single breakthrough, like those have been solved. And kind of what's left are these like fundamentally, inherently multivariable problems that there will never be like a single breakthrough to cure them. It's going to have to be some combination of humans and AI working beyond what humans are capable of actually like in their own brains synthesizing as a gestalt moment, as an aha moment.
That's interesting. I mean, I would not agree with that, actually.
That's why I asked you.
Yeah. So I still think that a huge issue now is what we're seeing is we have this ability to generate these big data sets with our next generation sequencing through other technologies. I think AI, whether, and again, and typically I would say the kind of like machine learning, deep learning type approaches are being applied more widely.
Right.
But the issue is that they're going to come up with these signatures that need so much additional validation. You still need the human insight to say, which is the thing that I am going to pursue and actually try to validate this.
Yeah.
And I think that's really still the challenge. So I agree. I think it's going to be a tool, but I haven't seen anything to say that AI is going to replace that aha moment. Because I think that AI might give you one additional piece of information you're going to fit into to say, this is the insight I think is actually really important. Right. But it's still going to be a huge investment to actually show something really worse in patients, show something really worse to treat disease. And I still think that decision-making process is going to be hard to replace.
Yeah, and I don't think she's, and I'm probably misquoting her, I don't think she was saying that there's no role for human creativity here, just that it is no longer going to be sufficient. The creativity has to come from a different sort. In a different field, so Garry Kasparov, the chess master, spent most of his life saying that, computers will never beat people at chess. Like, they just made this blanket statement. I remember when I was like 8 years old, I read the statement and kind of said like, I don't think you understand what computers are. And then, of course, he was beaten by Big Blue, by IBM, I think in the 80s. And but then, he formed this new theory, which has also since been disproven, which is kind of a shame because it's an elegant theory. But he said the future of chess is, he called it like centaur chess or something, where it's like a computer plus a human grandmaster playing another computer plus a human grandmaster. And he's saying that if you have a computer on both sides and a human on both sides, that will always be the superior game because the computer will prevent you from making any stupid mistakes, but all of the creativity and the strategy still comes with the humans. And indeed, for 20 years or so, the best chess matches were always computer plus person, always beat just a computer or just a person. More recently, I think that's actually been disproven. I think actually now the human is a handicap where just a computer does better when it doesn't have to justify itself to a human. Where do you think we are kind of on that spectrum for what you're working on? What is the role of the human intuition?
Yeah, no, I agree. Well, I think one area where this is very fraught based is for me as a pathologist, so in my clinical training. So I think one of the most exciting advances in terms of kind of AI in medicine is actually automated image analysis for pathology, because something we think about a lot because essentially not my part of pathology is focusing on kind of clinical lab diagnostics, but what most people do is you take a piece of tissue from a human, so a biopsy sample, an organ that's been resected in a surgery, and you basically cut it up, put it in slides, and look it under the microscope, and you make a diagnosis. Yeah. And so, but one of the big questions, and I think people in the field have been afraid of, to be frank, is that computers going to be able to do this better, more robustly, faster, more accurately. And so I think that is an area where the human may end up being a handicap, but maybe we're going to end up at the point where, these software are going to be pretty good. They're going to be important decision support tools, but you still need a human there to kind of make a final decision. I think the one thing that's going to protect pathologists, honestly, at least, is our kind of medico-legal framework, is that you need a person to sign off on things and someone to take legal liability. So you still need someone to do that. But, but I think when it comes to the automated image analysis, I would say too, especially the place that's been the most advanced is radiology, because that's kind of easier kind of, I think, in terms of the data that you get, it's easier to make progress there in terms of building data sets and kind of some of the classifiers that have been made. But already applications to radiology residency are down quite a bit because people are worried about just the computer's going to take all the jobs. So it doesn't make sense to be a radiologist.
Yeah. And interesting. And of course, diagnosing a cancer or even proposing a treatment is higher stakes than winning a game of chess. So you can see maybe the human intuition and the fail-safe playing a bigger and bigger role there, especially as we get into the even more controversial areas like gene editing, like CRISPR, which is, I know, something that's been applied to cancer research quite a bit. What do you think is the role of gene editing for cancer treatments?
Oh, I mean, I think it has a lot of potential. I think the big advances that we've seen so far, in terms of the first kind of CRISPR or other gene editing technology that's really gotten into patients, is basically, and something that we're working on in the lab now, is trying to make engineered T cells to fight cancer. It's a really huge burgeoning field. We're definitely not one of the first to be in it by any means. But this is one of the things that's exciting, is people have realized that not only can we take T cells, so an immune cell type from a patient, take them in the lab, engineer them so that they can fight cancer by basically making them express this gene and this protein they wouldn't normally express. But then the other option is, can we use a CRISPR-based approach to delete genes that we think might have a bad function, basically, in terms of making them, say, what we call exhausted. So basically making them so they stop functioning as well, fighting cancer so early. Can we do things to delete genes so they evade the human immune system better and they can persist for longer? Because it's one of the problems we realize, these cellular therapies have become very powerful, but part of the problem is that they don't last forever. And so can we do things to make them last longer? So I think in that context, that's where we've seen things go into patients right now, where we can basically delete these genes, try and make them last longer. Clinical trials are still going on, so we don't know if that's going to work yet. I think the other big advance that's going to happen is using CRISPR similarly to edit cells that you can implant into anyone. So this is a big strategy is so-called allogeneic CAR Ts, which basically means right now when we make these CAR T cells, these engineered T cells, we have to take T cells from a patient, ship them to some remote lab. Mm-hmm. They engineer them, send them back to the hospital, and they have to get reinfused back into that same patient. But you can't use it for anybody else. The big hope is that we can make a type of cell that they could just make in one central facility, and you could just buy it from the pharmacy, a hospital pharmacy. You wouldn't go to Walgreens, but you would have it in the hospital formulary, be able to give it to anybody. And so I think that's another place where CRISPR is really going to make a really big advance.
Where do you think the safety rails need to be on something like this? I think in any kind of public or even relatively well-informed kind of conversation of CRISPR, there's always there is the excitement of what it could do, and then there's the excitement of what of what it shouldn't do, of how it could really break things pretty fundamentally.
Well, I think that's what we're seeing now in terms of the first approaches, and I'd say still semi-tentative, and there's a lot of regulatory issues. Like FDA has been very hesitant, Food and Drug Administration, who regulates all of this, has been very hesitant to allow these trials. There's actually been a lot more done in China because they're a little more cavalier about these things. Yeah. And so they're actually far ahead in terms of clinical trials. But, I think that one of the big concerns has been, could these cells that are edited, one issue with CRISPR is that we say we want to edit this one specific part of the genome, but it might cause changes in other places. So that's been the main thing that people have been worried about is, could these cells on their own somehow turn cancerous? That's kind of been, or could they cause some very severe autoimmune disease where they attack other parts of the body besides the cancer? Especially
if they can multiply on their own after the fact. And so I think there's some proposed constraints about not being allowed to engineer CRISPR cells that are self-replicating.
I think that's pretty tough, at least in this cellular cancer therapy space. I think that would be difficult in the sense that you kind of need them to proliferate and replicate. Exactly. They need to be doing. So one thing a lot of people do know, though, is you can put in a kill switch. So basically one strategy has been to, when you're putting in that gene that causes the T cell to kill cancer, you also put in another gene at the same time that gives it a way you can kill it, either via a small molecule. So some use an approach where you give this standard antiviral drug that can kill the cells. Another one might be you give a different cancer therapy, the so-called monoclonal antibody, that then can deplete those cells and take them out. So I think that's one thing that's been put in place. I think the bigger question still, though, is, I think some people raise reasonable arguments, though, about there's so much hesitancy about the so-called potential for tumor development or other issues. But if you're giving these to a patient who's already dying of cancer, maybe it's not that big a deal. Maybe we don't need to, maybe we don't need to put the brakes on as much as we have been. When you come to other diseases that maybe are not as deadly or longer term, then maybe you need to have more safety regulations in place. But maybe we put them in the cancer patients earlier, get more data, see if any of these potential or theoretical outcomes come to pass, but maybe it's not that big a deal.
So what are you kind of most excited about? And what's conversely, what's overhyped in the industry? Because there's a ton of hype about basically every, you go to Y Combinator demo day, and literally, I'm going to try not to exaggerate, I think there's 200 something startups every demo day, and at least a third of them are curing cancer. Any given season, at least a third of them are it's something AI, something cancer, blockchain, something. Give us money. Where's the hype, and what are you excited about? And hopefully some of the stuff you're excited about is the stuff you're actually working on. So also, what are you actually working on?
No. I think one caveat, talking about the cellular therapy side, I think there is a lot of hype around that. And again, that's not, most of it I'd say is not necessarily involving AI right now in terms of cellular therapy side. But I think there is a big caveat. Maybe these aren't going to be that great in the end. I think we still don't know if we're going to be able to have persistent therapies that cure cancer all the time. It might be better to have other, so-called biologics, like antibody-based therapies that maybe they don't cure the cancer right up front. But again, that you can have off the shelf, you can give to anybody, they'll be a lot cheaper. I think that that's a big caveat in the field. When it comes to, you're right. When it comes to kind of AI-based approaches in medicine. I think one thing that we've seen recently is kind of the failure of Watson, basically, that had all this hype, and they were, IBM was going to revolutionize everything, and Watson was going to go through a zillion research articles and propose new treatments for your patients. But it turned out not to come to pass, basically. I mean, at least I heard they had a big trial with Memorial Sloan Kettering, and no one thought it did a good job. And so I think that
I think it turned out that Watson was, marketing.
Yeah, exactly. So I think that's the issue, is that when you just because you have a big data set and because you're able to build some way to interpret it doesn't necessarily mean that it's going to have those effects on patient outcomes. And so I think that's still the main question is, can you get too far ahead of yourself with your technology and not necessarily be able to come back and show that it actually has a relevant application, basically? Or do you have a technology in search of a question?
Are you comfortable discussing maybe a thing that you're excited about that's coming out of your lab, or you don't have to if you don't want to?
No, it's something that our kind of first foray, I'd say, in terms of machine learning. We collaborated with this group at Penn, basically, where in the blood cancer we study multiple myeloma. There's a gene called RAS, and it's kind of the most common bad player in all human cancers. It's the most commonly mutated cancer. There are certain mutations we know that really drive tumors, and it's not just in blood cancers, but it is in many blood cancers, but also solid tumors. And so what we did is we built a classifier looking at all the genes expressed within the cells that had two different types of these mutations. Most people treated them the same, but we could find that these are actually quite different. And then we looked further into it and see that the patients with these different mutations have very different outcomes, and we proposed that you actually want to treat them in different ways. So this is a hypothesis we could build based on an AI type approach. But the issue is that now the next step is really demonstrating that in patients.
Right.
And so the question is, who wants to invest and put that in a clinical trial? And so I think that's where we're going to see a lot of these, these same questions arise. But, in terms of what we're working on my lab overall is, we're really interested in using mass spectrometry-based proteomics. Again, another big term, but essentially what it allows us to do is look at changes in hundreds or thousands of proteins within cells. We're looking in the context of resistance to these cellular therapies, because patients get treated with them, but they don't always respond. Or maybe they do respond and then the cancer comes back. We're trying to understand what's changing within the cells at the level of their proteins. Can we come up with new targets that are new vulnerabilities in the resistant cancers? So that's a big thing that we're trying to do.
Very cool. Well, thank you, Arun, for coming in. We do a lot of talking with people that are working on the kind of cutting edge of what's next for healthcare and for medicine. And it's super gratifying to hear what you're working on.
Cool. Thanks so much for having me.
Thank you.
And now it's time for a listener question, and this one comes in from Bill via Twitter. And Bill says, does All Turtles consider the Illuminatus trilogy to be sci-fi? And I suppose this is in response to the, was it an 8-part sci-fi partnership series this past summer? Good question.
It is a good question. It's been a while since I read the Illuminatus trilogy. That's by Robert and Robert, right? Like Robert Wilson and Robert somebody.
That sounds right.
Which is awesome. It's a great book. No, it's not sci-fi. It's totally true. It is investigative journalism. I don't know. Are we perilously close to getting into QAnon territory here? I don't know. I think the Illuminati trilogy is super cool. It may be more like parody or comedy than sci-fi.
I think fantasy.
Yeah,
fiction
maybe fantasy. But it's definitely worth reading if anyone hasn't picked it up. And it is sadly being taken more seriously now than it I think was intended to be. But it is a cool book. But yeah, as John was saying, we did just record a whole series on sci-fi entrepreneurship, how sci-fi informs entrepreneurship and what we build. Some really cool interviews. Brittany Gallagher, my co-host, and I interviewed a bunch of really fascinating people about
really
sci-fi.
Cool.
So you guys should check that out. We talk about stuff that is definitely straight up influential sci-fi.
What's the difference between sci-fi and fantasy-fi? One's more magic. One's more kind of tech dystopian.
Yeah, one's got elves, one's got space elves. I think while they do come together quite a bit, to me the difference is I'm a huge fan of both. I've read all of Tolkien and everything else. To me, compelling sci-fi is about world creation, and as is compelling fantasy. They're both like painting a plausible world That's kind of different from our world in some ways. But with sci-fi, it's a world that we can envision how we would get to from here. Where fantasy, it's usually kind of a compelling world. It's a fantasy of something that may have been in the past. So for me, the kind of sci-fi that I really like are the world building sci-fi, which is here is how we could plausibly get to the world that's presented in the work. Whether we want to or not is a separate question. That's in fact a big part of what we talked about in Sci-Fipreneurship, the series, is how many of these projected worlds are dystopian, even though some of the technology that's presented in them are kind of cool. I don't think that like the Lord of the Rings reads as like, it's like a very compelling world, but it's not a world that's like seems like we're heading in that direction.
Right.
As opposed to a Dune or 1984 or any of these things that could be.
Cool. Well, thanks, Bill. We appreciate the question. For any other listener questions, you can email us at hello@all-turtles.com or tweet us at @allturtlesgo. And Marie will most likely read it.
This podcast is a production of the All Turtles Worldwide Media Empire. We recorded this episode in the world-class Donatello Studios in San Francisco, California. Thanks to Arun Wiita for joining us in this episode. Thanks to everyone who made this episode possible, including Jem Metzendorf for editing, Marie McCoy-Thompson for producing, Chris Plogue for his audio expertise, Mike Rivera for our artwork, and Matt Ammerman for our theme music. On behalf of Jessica Collier, John Cifuentes, and yours truly, Phil Libin, and all the rest of the All Turtles team, thanks for listening.